what if we could make living tissues, organs, as we make 3D objects? Could, with this capacity, reduce or contribute to reduce organ shortage? Because you probably know that there is a huge demand for organ transplantation, and that this demand is continuously increasing in Europe, in the US, and all over the world. Could, with this capacity, could we reduce the number of animals that are killed for drug testing, for instance? This is, all over the world, a huge trend by a lot of people willing that animal testing is reduced at the maximum to test drugs or even cosmetics products. Also, could we imagine, thanks to printing organs or tissues, that we will be able to speed drug development and to reduce the cost of drug development by developing some more predictive tools that will allow pharmaceutical companies to test more efficiently drug candidates. So this is what we think at Poeitis. We do think that bioprinting, printing tissues, and in the next future, more complex tissues and eventually organs will be possible. So Poeitis, it's a spin-off of INSERM, that is the National Institute for Health and Medical Research in France, probably the largest institute for medical research in Europe. We were founded in 2014, and we are almost more than 20 now. And so far, we have been able to win some different challenges, like the World Innovation Challenge last year and this year again. So probably one of our key milestones were to be able to partner with some large companies like L'Oréal and BASF. So what is this technology? This technology is bioprinting. It's a combination of 3D printing and biology. So from 3D printing, we take the capacity to deposit, to create 3D object by depositing layer by layer from a 3D file. And from the biology, we take all the biological laws that drive tissue growth and morphogenesis. So we use to describe bioprinting as a sequence of 5 different steps. First, I would say the first step is inspiration. We need to look at how the biology, how the tissue, how the organs are made, and for that we use imaging tools that come from the medical side or that come from laboratories like confocal microscopy. From this microscope, we can define the organization of cells, of tissue component into 3D. And for that, we use computer-assisted design software that allows us to position this tissue component into 3D. So we obtain a 3D file, which is then sliced, cut into 2D files that are afterwards print layer by layer to create a 3D structure. What is really important in the context of bioprinting, as compared to conventional 3D printing of metals or polymers is that after printing, the object evolves with time. That means that cells interact with each other, and cells interact with their environment. So the final produce is really different from the produce or the object, the structure that is obtained just after 3D printing. So to do that, we have developed for the last 10 years, at the beginning at INSERM and at Poeitis, a specific technology which is laser-assisted bioprinting. So it works like an inkjet method, meaning that we have a laser pulse instead of a thermal pulse that is focused on a glass slide that is covered by a liquid film that contains cells. And by focusing the light at the interface, we can propel some very small jet and then collect very small droplets onto a substrate that, on these droplets, contain a very precise number of cells. So laser-assisted bioprinting has the highest resolution of all bioprinting technology. This technology provides the capacity to print with single-cell resolution. That means printing cell by cell. And thanks to this capacity, we are able to standardize tissue manufacturing process. That means that we are able to control precisely the number of cells into a 3D construct and the position of all these cells, which is critical for the next application, I mean clinical application. Interestingly, this high resolution allows us to control morphogenesis process as well. Morphogenesis is a tissue formation process. So, and we have shown that depending on the initial cell organization, we can observe after time, and I will show you later some example. After time, we can observe some specific organization, shape, and function. Finally, thanks to this high resolution, we can produce highly complex structure. And interestingly, we can consider this example of the hair follicle. We have this project with L'Oréal of printing hair follicle, which is a really complex organ made of about more than 10 different cell types organized in a very, very small space of about 0.5 millimeter. So to position all these cells into such a teeny environment, it was necessary to have a high-resolution bioprinting technique and so the capacity to print with single-cell resolution. So to wrap up, thanks to the high resolution, we will be able to ensure the manufacturing of safe tissue that is critical for clinical application, to improve tissue functionality the functionality of tissues, and also to develop some patient-specific tissues. So during the first years of the company, we have developed a new bioprinter that you can see on your right. So this bioprinter integrates a lot of modules, including it's a multimodal system with sensors and so on, a lot of tools to control the bioprinting process. And on the left, you can see our new computer-assisted design software that we developed. So far, no CAD software was developed for bioprinting application. So as you can see, with this tool, we can position virtually all tissue components into 3D and generating with these tools 3D files that is exported to the bioprinters afterwards. So thanks to these bioprinting tools, we have been able to print some 3D structure. As you can see on this movie, we can print and deposit very precisely different layers of cells with high resolution and high reproducibility. And on your left, what you can see also is that the cell pattern after 2 days is different from the initial. That means that we need to take into account how cells pattern evolve with time. So basically, this gave birth to the concept of what we call 4D bioprinting. 4D bioprinting means to program tissue self-organization. That means to design an appropriate 3D organization of tissue components, of cells, of extracellular matrix proteins, for instance, so that at the end, after maturation, a specific tissue function emerges. Interestingly, we have shown that depending on the initial position of fibroblasts, cells that are included into the skin dermis, we can control how these cells remodel, reorganize the extracellular matrix, and namely collagen fibers. This is really interesting because that allows us now to produce some skin dermis equivalent with some interesting properties. So this is one of our main results so far that we obtained with our technologies. This is a skin, a fully bioprinted skin. Typically, it's a piece of few square centimeter of skin made of both a dermis and an epidermis. So the dermis is made by printing both collagen and fibroblasts, and the epidermis is made by printing keratinocyte onto the top of this structure. What we have shown is that after printing, we observe an histoarchitecture, a tissue organization that is quite close from native skin, and we also observe that after time, the keratinocyte differentiate accordingly to a mature epidermis. With one of our partner, BASF, we also developed another skin tissue model, even a bit more complex. As you can see on this video, you can see the different layers of the skin from the top of the epidermis to the bottom of the dermis, and you can also see the organization of collagen fibers that show how functional bioprinted tissues are thanks to our technology. So finally, today we are able to print tissues, eventually simple tissue like skin. We hope to be able to implant these tissues into humans into the next years. Our goal is to implant such kind of skin, bioprinted skin, into human close to 2020. And hopefully we will be one of the first in the world to do that. But as for organ, probably I used to say that it's like, for us, it's like to go to Mars. We don't know if we will be able to go there. What we think is that to go there, to be able to print such complex structure, which are organs, we will need like a transnational program with a strong collaboration with physicists, clinicians, and people on some companies to be able to create liver, kidney, or even heart. So if you want to join us or participate to our crowdfunding campaign, because this one is closing next Sunday, do not hesitate to contact us and to go to our website. Thank you very much.